Welcome to the Sleepy Mind channel. Tonight we are going to spend some time with fireflies. On warm evenings, as daylight fades and the air begins to cool, small points of light sometimes appear close to the ground. They rise slowly from grass, along hedges, and near the edges of trees. The light appears without sound and without any pattern that is immediately clear. Fireflies are beetles that produce light inside their own bodies. The light comes from a controlled chemical reaction that releases very little heat. It turns on and off with precise timing, guided by the nervous system and supported by oxygen moving through the abdomen. In this story, we will follow how that light is made, how it is used, and how it fits into the life of the firefly and the night environments where it appears. Let us begin, and if you enjoy these stories, please like and subscribe for more videos. As daylight fades, the environment begins to shift in ways that are gradual but measurable. Light levels fall first, then colors lose saturation and edges soften. The ground, warmed through the afternoon, releases stored heat back into the air. Air movement often slows, and temperature differences between shaded and open areas narrow. These changes do not signal inactivity. They mark the transition into a different working state of the environment. Night alters how space behaves. Without direct sunlight, surfaces stop reflecting strong glare. Instead, they absorb and release energy more evenly. Moisture becomes more noticeable as evaporation slows. Thin layers of humidity form close to the ground, especially in areas with vegetation or exposed soil. This creates a stable band of air where small organisms can move without rapid temperature loss. Sound travels differently once the atmosphere cools. With less turbulence, vibrations carry farther and remain clearer. At the same time, background noise drops as daytime activity ends. The environment becomes quieter, not empty. Small movements, wing beats, and contact with leaves become easier to detect. For animals active at night, this shift changes which senses provide the most reliable information. Light at night behaves as contrast rather than illumination. Even weak sources stand out against a dark background. Reflected moonlight outlines surfaces rather than filling space. A brief glint or flash becomes noticeable because there is little competing brightness. This makes darkness a condition where small visual signals can carry meaning if they are precisely timed and placed. Temperature stability is another defining feature. Once the sun sets fully, heating stops and cooling slows. The range between warm and cool narrows through the night. For small-bodied animals, this matters. Heat loss becomes more predictable. Movement requires less energy spent on regulation. Muscles can operate within steady temperature limits, rather than adjusting constantly. Plants contribute to this stability. Leaves and stems hold moisture and release it slowly. Vegetation reduces wind near the ground, creating sheltered layers of air. Chemical processes inside plants continue through the night, driven by stored energy rather than incoming light. Some plants increase scent release after dusk, changing the chemical landscape without changing the physical structure. The ground layer becomes a central corridor for activity. Leaf litter, soil particles, fallen wood, and low plants form a complex surface with many small shelters. Temperature here changes more slowly than in open air. Humidity remains higher. For insects, this layer offers protection and predictable conditions for movement. Insects that are active at night often time their emergence carefully. They respond to the rate at which light fades rather than darkness alone. As dusk deepens, Internal clocks trigger changes in muscle readiness and sensory sensitivity. Wings warm slightly through movement. Nervous systems shift into patterns suited for low-light operation. Fireflies emerge within this window. They do not appear as soon as the sun sets. They wait until contrast is sufficient and air temperature supports flight. Their activity is tied to specific thresholds. Too much light reduces signal clarity. Too little warmth limits movement. The night environment provides a narrow zone where both conditions align. Within this zone, space becomes readable in a different way. Open areas allow light to travel horizontally near the ground. Edges between forest and field form corridors where signals can move between vegetation gaps. Moist areas scatter light slightly, shortening range but increasing softness. Each habitat shapes how far a flash can be seen and from which angles. Movement at night often slows. This reduces energy use and lowers detection by predators that hunt through motion. Fireflies tend to fly low and steadily, often pausing on stems or leaves. Their light is not constant. It appears briefly, then stops. This pacing matches the environment, where pauses allow signals to stand out without creating continuous exposure. The night environment also reshapes risk. Some predators learn to associate light with prey. Others rely on sound or vibration. Fireflies navigate this by limiting flash duration and spacing signals over time. Darkness allows this restraint to work. In bright conditions, the same signals would lose distinction or draw too much attention. Energy use becomes more tightly managed at night. Flight, signaling, and basic metabolism all draw from stored resources. Fireflies benefit from the efficiency of their light production, which releases very little heat. Even so, each flash requires oxygen delivery and precise control. The stable temperatures of night help keep these processes reliable. Over many generations, night conditions have shaped bodies and behavior together. Sensory systems favor timing over detail. Nervous systems refine control over short bursts of action. Chemical pathways operate within narrow ranges that match evening temperatures. The environment does not demand speed, it demands precision. As full darkness settles, the night reaches a steady state. Background light remains low. Temperature changes slow further. The air near the ground stays calm and moist. In this setting, small changes become noticeable. A brief pulse of light can mark presence, direction, and movement. The night functions as a medium that carries information quietly. This is the environment fireflies depend on. Darkness here is not absence. It is a condition shaped by cooling air, stable ground temperatures, and high contrast. Within it, light produced by a living body can be seen clearly, timed carefully, and used without waste. The night provides the space where such signals make sense and where the firefly's life can unfold. Darkness does not arrive as a single condition. It brings a set of limitations that change how living systems function. As light levels fall, the amount of information available to the eye decreases. Colors fade first, then fine detail. Depth becomes harder to judge, and movement blends into background shadow. For organisms that rely on vision, these losses immediately affect how safely they can move. When visual detail drops, bodies adjust. Movement slows because obstacles are harder to detect. Paths that were easy to cross during the day require more caution. Animals often remain closer to familiar surfaces, using edges, ground texture, or vegetation as guides. This reduces the risk of collision and wasted effort. Darkness reshapes behavior by narrowing which actions are reliable. Hearing and touch gain importance as vision weakens. Small vibrations become more noticeable when background noise decreases. Contact with surfaces provides orientation. Antennae, whiskers, and sensitive skin regions begin to carry more information than distant sight. For insects, air movement and surface texture become critical signals that guide direction and speed. Chemical sensing also becomes more effective at night. Cooler air slows the breakdown of scent molecules. With less turbulence, chemical trails persist longer and spread more evenly. This allows organisms to follow gradients rather than fleeting signals. Many insects rely on this stability to locate food, mates, or shelter when light is limited. Darkness also changes how predators and prey interact. Visual hunters lose accuracy. Their reaction times increase as they struggle to judge distance and speed. Predators that hunt through sound, vibration, or chemical cues gain an advantage. Prey species respond by reducing unnecessary movement and choosing timing carefully. Survival becomes less about speed and more about control. Physiological processes shift under these conditions. Without solar heating, body temperatures stabilize within a narrower range. For small animals, this reduces the energy spent on cooling or warming. Muscles can operate more consistently. Water loss through evaporation decreases, which is important for insects with high surface area relative to volume. Internal clocks play a central role in these adjustments. As light fades during dusk, photoreceptors signal changes to the nervous system. Hormone levels adjust gradually rather than abruptly. This prepares tissues for night activity by changing muscle readiness, sensory sensitivity, and metabolic rate. Timing becomes as important as capability. Plants contribute indirectly to these biological pressures. After sunset, photosynthesis stops, but respiration continues. Water transport slows. Many plants increase scent release, which supports night-active insects. This shifts the sensory landscape from visual to chemical without altering physical structure. Darkness encourages communication that does not rely on sight. The structure of the environment amplifies these effects. Open areas allow limited light to travel farther, while dense vegetation absorbs and scatters it. In cluttered habitats, visual range shortens further, increasing reliance on close-range cues. Darkness does not affect all spaces equally. It creates gradients of usability across the landscape. For insects, these gradients determine where activity concentrates. Paths, clearings, and edges become preferred routes. Areas with stable humidity and temperature offer safer movement. Night selects for behaviors that match these small-scale differences rather than broad visibility. Fireflies are shaped by these same pressures. Their signaling system depends on contrast. When background light remains too high, flashes lose clarity. When light drops too low or temperatures fall too far, flight becomes inefficient. Fireflies therefore operate within a narrow window where darkness limits vision but still supports movement. Producing light is costly. Even with efficient chemistry, each flash requires oxygen delivery, nervous control, and chemical fuel. Darkness restricts waste. Signals that are unclear require repetition. Repetition increases energy use and exposure to predators. Over time, fireflies that flash with precise timing and spacing are more likely to persist. Darkness also limits competition. Many diurnal insects retreat into shelter, reducing crowding. Resources that were contested during the day become available. Flowers that release scent at night face fewer visitors. Fireflies benefit from reduced interference when locating one another through light signals. The night environment encourages restraint. Continuous activity becomes risky and inefficient. Short movements followed by pauses reduce detection. Brief signals separated by silence stand out more clearly than constant output. Darkness rewards systems that can start and stop processes quickly without overshoot. These pressures act together. Reduced vision, stabilized temperature, persistent chemical cues, and altered predation all narrow the range of effective strategies. Bodies that depend on constant visual correction struggle. Systems that rely on timing, efficiency, and controlled output function more reliably. Over generations, anatomy and behavior shift in response. Sensory systems emphasize timing sensitivity. Nervous systems refine control over brief actions. Metabolic pathways adjust to support intermittent effort rather than continuous output. Darkness shapes biology by favoring precision over excess. In this environment, light produced by an organism becomes useful only when it is controlled. The night does not support constant display. It supports signals that appear briefly, carry clear information, and then disappear. Fireflies exist within this balance, shaped by the limits darkness imposes and the opportunities it creates. Bioluminescence begins as a physical process inside living tissue. Energy stored in molecules is released through a chemical reaction that produces light instead of heat. This reaction does not occur by accident. It requires specific compounds, precise timing, and controlled access to oxygen. Without these conditions, the reaction does not produce visible light. In the natural world, most energy release inside cells appears as heat. Muscles warm as they contract. Chemical reactions inside organs raise temperature slightly as molecules break apart and reform. Bioluminescence follows a different path. The energy released is channeled into photons rather than thermal motion. This makes the process unusually efficient and useful in environments where excess heat would be harmful. The origin of this process lies in chemistry rather than behavior. Certain organic molecules can exist in high-energy states. When they return to a lower energy state, the excess energy must be released. In most reactions, that release is spread across surrounding molecules as heat. In bioluminescent reactions, the energy is transferred directly into light. This transfer requires a precise match between molecules. One compound stores the potential energy. Another controls the reaction rate. Oxygen participates as a trigger rather than a fuel. Together, these components allow energy to be released in a short burst that produces visible light without raising surrounding temperature. The earliest bioluminescent systems likely evolved in environments where light carried advantage under low visibility. In dark habitats, even faint light can travel farther than chemical or mechanical signals. Organisms that could produce controlled light gained a way to mark presence, movement, or interaction without relying on sight in daylight. At first, light production may not have served communication. It may have begun as a byproduct of detoxification or metabolic regulation. Some chemical reactions that remove reactive oxygen compounds can release energy. When that energy is channeled through light-producing molecules, a faint glow can appear. Over time, systems that produced usable light gained additional function. Once light became visible outside the body, control became important. Continuous light would waste energy and attract attention. Intermittent light could be timed to specific moments. Organisms that developed ways to start and stop the reaction gained an advantage. Control turned light from a chemical side effect into a biological tool. Fireflies inherited this controlled system. Their light production occurs in specialized cells that contain the necessary molecules in high concentration. These cells are supplied with oxygen through a network of fine tubes rather than blood vessels. This allows oxygen delivery to be regulated rapidly, turning the light on and off within fractions of a second. The physical layout of these cells matters. Light-producing cells are layered with reflective structures beneath them. These structures direct photons outward rather than inward, increasing visible output without increasing chemical input. The body itself becomes part of the optical system, shaping how light leaves the insect. Energy efficiency is central to this design. Fireflies do not generate excess heat when they flash. This allows repeated signaling without overheating small tissues. In warm summer evenings, where ambient temperature is already high, this efficiency prevents damage and allows signaling to continue over long periods. The chemical pathway itself is tightly regulated. The light-producing molecule must be present in the correct form. The controlling enzyme must be active. Oxygen must arrive at the right moment. If any element arrives too early or too late, the reaction either fails or produces wasted energy. Precision is required at every step. This precision is supported by the nervous system. Signals from the brain control oxygen delivery to the light-producing cells. When nerves fire, oxygen floods the reaction site. When the signal stops, oxygen is withdrawn. The light appears and disappears in direct response to neural timing. Such control allows light to be shaped into patterns. Duration, spacing, and repetition become adjustable features. These features can carry information if they remain consistent. Over time, species that used light in predictable ways gained the ability to recognize one another through timing alone. Bioluminescence also appears in many unrelated organisms. Marine plankton glow when disturbed. Fungi produce steady light along growing tissue. Bacteria emit light continuously when chemical conditions allow. In each case, the same principle applies. Energy is released as photons through controlled chemistry. What differs is how that light is used. In the ocean, light can confuse predators or draw attention away from vulnerable tissue. In forests, light can mark position in dense vegetation. In insects like fireflies, light becomes a signal exchange between individuals moving through darkness. The origin of bioluminescence is therefore not a single event. It is a repeated solution to the same constraint. In dark environments, communication and detection become difficult. Chemistry that allows energy to exit the body as light offers a way to overcome that limit. Fireflies represent one of the most refined versions of this solution. Their system produces bright light with minimal waste, responds quickly to neural control, and integrates with body structure to maximize visibility. The light they produce is not decoration. It is the visible outcome of chemistry shaped by physical limits and environmental need. This chemistry sets the foundation for everything that follows. Without efficient light production, signaling would not be possible. Without control, signaling would not be reliable. Bioluminescence provides the mechanism that allows fireflies to operate within darkness, using light as information rather than illumination. The chemistry that produces light in fireflies operates inside specialized cells located near the end of the abdomen. These cells contain the materials required for light production and are arranged to allow rapid control. When a firefly flashes, nothing external ignites. The reaction happens entirely within tissue that is prepared for this purpose. At the center of the process is a small organic molecule called luciferin. Luciferin stores chemical energy in its structure. On its own, it remains stable. It does not glow spontaneously. To release its energy as light, it must enter a controlled reaction sequence that changes its chemical state. That control is provided by an enzyme known as luciferase. Enzymes act as biological tools that guide reactions without being consumed. Luciferase binds to luciferin and positions it precisely. This positioning allows the reaction to proceed quickly and cleanly, rather than releasing energy slowly or unpredictably. Oxygen plays a critical role at this stage. Oxygen enters the reaction site through a network of fine tubes called tracheae. These tubes deliver air directly to tissues without using blood. In the light-producing cells, oxygen arrival is tightly regulated. When oxygen concentration rises, the reaction proceeds. When it falls, the reaction stops. The sequence begins when luciferin binds to luciferase. Energy stored in cellular molecules prepares luciferin for oxidation. Oxygen then reacts with the prepared luciferin. This reaction raises the luciferin molecule into an excited state. In this state, electrons occupy higher energy positions than usual. Excited states are unstable. The molecule cannot remain in this configuration for long. As electrons return to their normal positions, the excess energy must be released. In bioluminescence, that release occurs as a photon of visible light rather than as heat. The color of the light depends on the exact energy change involved. In fireflies, the emitted light is typically yellow-green. This wavelength travels efficiently through humid night air and is easily detected by the eyes of other fireflies. Producing this specific color is not accidental. The chemical structure of luciferin and the environment created by luciferase shape the energy release precisely. Heat production during this process is minimal. Most chemical reactions inside living tissue release energy as heat, which spreads into surrounding cells. In fireflies, nearly all the energy exits as light. This high efficiency protects delicate tissues and allows repeated flashing without overheating. Control over oxygen flow determines when the reaction occurs. Fireflies do not store oxygen inside the light-producing cells in large amounts. Instead, oxygen arrives when nerve signals open pathways that allow air to reach the reaction site. When the signal ends, oxygen levels drop quickly and the reaction stops. This start and stop control allows flashes to be brief. A single flash may last only fractions of a second. Between flashes, the reaction components reset. Luciferin returns to a stable form. Luciferase remains ready for the next cycle. Oxygen levels fall to baseline. The system waits. The physical arrangement of the light organ improves efficiency further. Beneath the light-producing cells lies a layer of reflective tissue. This layer contains crystals and structures that reflect light outward. Photons that would otherwise be absorbed by internal tissues are redirected toward the exterior. Above the light-producing cells, the body wall is thin and lightly pigmented, This reduces absorption and scattering. The abdomen becomes a window through which light exits with minimal loss. The firefly's body therefore acts as both a chemical reactor and an optical device. The nervous system coordinates this process. Electrical signals travel from the brain to the abdomen along nerves. These signals do not directly trigger the chemical reaction. Instead, they regulate oxygen delivery by controlling muscles that open or close air passages. Timing at the neural level becomes timing at the chemical level. Because neural signals can be patterned precisely, flashes can be patterned as well. Duration, spacing, and repetition all depend on how long oxygen is allowed to reach the reaction site. Small changes in timing produce noticeable changes in visible output. This allows light to be shaped into consistent sequences. The chemistry itself does not carry meaning. It produces light whenever the conditions are met. Meaning emerges when timing becomes consistent across individuals. When many fireflies of the same species produce similar flash patterns, the light becomes recognizable rather than random. The same chemical principles appear in other bioluminescent organisms. Marine species use related luciferins and luciferases. Fungi produce light through slower, continuous reactions. Bacteria glow steadily when chemical conditions allow. In each case, the reaction follows the same physical rules. What differs is control. Fireflies represent a system where chemistry is placed under rapid neural regulation. This allows light to appear only when needed and disappear just as quickly. Such control is rare and depends on precise integration between chemistry, anatomy, and nervous system function. The chemistry behind living light therefore operates as a chain of physical events. Molecules bind, oxygen arrives, energy shifts, light exits the body. Each step must occur in the correct order and within narrow limits. When those limits are met, light appears without heat and without damage. This chemical foundation allows fireflies to use light repeatedly and reliably. It supports signaling that can occur night after night across a brief adult lifespan. Without this controlled chemistry, light would be either wasteful or uncontrollable. The reaction provides the mechanism that makes biological light usable in a dark environment. Fireflies belong to a group of insects defined by their body structure rather than their light. They are beetles. This means their bodies follow a specific physical plan shared by many insects that do not glow. Understanding this structure explains how fireflies move, feed, and protect themselves before light ever becomes part of the picture. A beetle body is divided into three main sections. The head contains sensory organs and mouth parts. The thorax supports movement. The abdomen houses digestion, reproduction, and in fireflies, light production. Each section is built from hardened plates, joined by flexible membranes. This combination provides protection without preventing motion. One defining feature of beetles is the presence of hardened front wings, called elytra. These wings do not function in flight. Instead, they act as protective covers. When a beetle is at rest, the elytra close over the softer hind wings and abdomen. This shields delicate tissues from abrasion, moisture loss, and predators. In fireflies, the elytra are thinner and more flexible than in many ground-dwelling beetles. This reflects their need for flight. When a firefly prepares to fly, the elytra lift outward and upward. Beneath them, a second pair of wings unfolds. These membranous wings provide lift and propulsion. The elytra remain raised during flight, acting as stabilizers rather than flight surfaces. The thorax contains the muscles that drive this movement, large muscle groups attached to internal supports rather than directly to the wings. When these muscles contract, they deform the thoracic structure slightly. This deformation causes the wings to move. The system allows rapid, repeated wing beats without direct muscle attachment to each wing. Beetle legs also reflect their structural identity. Fireflies have 6 legs attached to the thorax. Each leg ends in small claws that allow gripping stems, leaves, and uneven surfaces. The legs are not built for speed or jumping. They are built for steady movement and secure attachment. Fireflies often pause while signaling, and their legs allow them to remain anchored during these periods. The head of a firefly contains compound eyes adapted for low light conditions. Each eye is made of many individual units that detect changes in brightness rather than fine detail. This suits night activity, where contrast matters more than sharp outlines. The eyes are positioned to give a wide field of view rather than precise forward focus. Antennae extend from the head and serve multiple functions. They detect chemical cues in the air and help sense nearby objects. In some species, antennae are more elaborate, increasing surface area for chemical detection. These structures assist in locating mates and navigating complex environments where vision alone is limited. Mouth parts in adult fireflies are relatively simple. Many species feed little or not at all during adulthood. Their primary energy reserves were accumulated during the larval stage. As a result, adult mouth parts are not specialized for heavy feeding. This reduces weight and allows more energy to be allocated to flight and signaling. The abdomen remains flexible despite its segmented appearance. This flexibility allows expansion during breathing and movement. In fireflies, the abdomen also houses the light-producing organs. These organs occupy specific segments near the tip. Their placement ensures that light is visible during flight and when perched. The beetle body plan also includes a tracheal breathing system. Air enters through small openings along the body called spiracles. From these openings, air travels through branching tubes that deliver oxygen directly to tissues. This system allows rapid oxygen delivery without relying on circulation. In fireflies, this is essential for controlling light production. Protection is another benefit of beetle structure. The hardened exterior reduces water loss and provides defense against minor damage. Fireflies often move through dense vegetation. Their bodies can withstand contact with stems and leaves without injury. This durability supports repeated nightly activity over the adult lifespan. Classification as beetles places fireflies within a large and diverse group. Many beetles are active during the day, others live underground or inside wood. Fireflies represent a lineage that adapted this basic structure to life at night and to the use of light. The underlying body plan remained the same. The specialization came through modification rather than replacement. This continuity matters. Fireflies did not evolve as glowing organisms first and beetles second. They are beetles that added light to an existing framework. Their muscles, wings, legs, and breathing systems all follow beetle design rules. Light production fits into this structure rather than reshaping it entirely. Understanding fireflies as beetles helps explain their limits. Their flight speed is moderate. Their movements are deliberate rather than fast. Their signaling occurs within short ranges, shaped by wing strength and visual capability. These traits reflect the physical boundaries of the beetle body. The beetle structure also shapes behavior. Fireflies often fly low and pause frequently. Their bodies support controlled movement rather than rapid pursuit. This matches a signaling strategy based on timing rather than chase. The structure and the behavior reinforce one another. Fireflies therefore carry their light on a body built for protection, steady movement, and efficiency. Their glow sits within a framework shared by millions of other beetle species. The difference is not the body itself, but how that body has been adapted to function in darkness, using light as a signal. The light organs of a firefly sit within the last segments of the abdomen, built from layers of specialized tissue that handle chemistry, oxygen delivery, and outward emission. When a flash occurs, a small region of the abdomen changes from dark to bright in less than a second. That change depends on physical structures that bring reactants together and remove them again without delay. The abdomen is made of repeating segments joined by flexible membranes. Hard plates on the upper surface protect the body, while softer tissue beneath allows expansion during breathing and movement. Near the underside of the final segments, the cuticle becomes thinner and lighter in color. This reduces absorption and allows more light to pass through. Inside these segments, the light organ is organized into distinct layers. The outer layer contains cells where the light-producing reaction occurs. These cells hold luciferin, luciferase, and supporting molecules in high concentration. They also contain organelles that supply energy to prepare luciferin for oxidation. The reaction remains quiet until oxygen arrives. Beneath the light-producing cells sits a reflective layer. This tissue contains dense granules and crystals that bounce photons back toward the outer surface. Light that would otherwise enter deeper organs is redirected outward. The effect increases brightness without increasing chemical fuel use. The abdomen becomes an efficient lamp shaped by biological material. Oxygen reaches the light organ through the tracheal system. Air enters the body through spiracles along the abdomen and thorax. From each spiracle, tubes branch into finer and finer passages. Near the light organ, the smallest tubes press close to the light-producing cells, placing oxygen within short diffusion distance. The tubes deliver oxygen directly, without passing through blood. Control depends on how oxygen flow is regulated. In many insects, spiracles open and close rhythmically to reduce water loss. In fireflies, the timing of oxygen delivery to the light organ is adjusted so the reaction can start and stop quickly. When oxygen concentration rises at the reaction site, the excited chemical state forms and photons are released. When oxygen drops, the excited state no longer forms and light ceases. Nervous control coordinates these changes. Nerve signals travel to muscles that influence air movement and pressure within the tracheal network. Small shifts in pressure can push air into fine branches or allow it to drain away. The light organ therefore responds to neural timing through mechanical control of gas exchange. The visible flash matches the internal timing of oxygen availability. Circulation supports the light organ in a different way. Insects do not use blood to carry oxygen, but hemolymph moves nutrients, waste, and signaling molecules through the body cavity. The light-producing cells rely on steady supplies of metabolic fuel to maintain readiness. Waste products from repeated reactions must also be removed. Hemolymph flow around the organ maintains chemical balance without being responsible for the on-off switch. The nervous system also shapes the flash pattern by controlling duration and spacing. A brief neural burst produces a brief rise in oxygen and a short flash. A longer burst sustains oxygen longer and extends light output. Repeated bursts create rhythms. Because the chemical steps occur quickly, timing is limited mainly by oxygen delivery and withdrawal rather than reaction speed. Firefly eyes are adapted to read these patterns. Compound eyes detect changes in brightness and motion. At night, they perform best when contrast is high and background light is low. The flash color and intensity sit within the sensitivity range of the firefly visual system. This matching reduces the need for strong light and supports energy efficiency. The abdomen contains other organs that must coexist with the light organ. Digestive tissue, reproductive structures, and storage reserves share the same body cavity. The placement of the light organ near the tip of the abdomen separates it from much of the digestive mass and keeps the emitting surface unobstructed. Segmentation also allows the emitting area to remain stable while other tissues shift during movement. Heat management remains important, even with efficient chemistry. The reaction produces little heat compared with most biochemical processes, but the abdomen is small and tissues are thin. Efficient emission reduces the need for long flashes that would increase metabolic demand. Short, controlled flashes keep energy use low and prevent local warming. The cuticle above the light organ is supported by microscopic structures that influence how light exits. Surface texture can scatter light slightly, producing a soft glow rather than a harsh point. This reduces sharp glare and can improve visibility through vegetation by spreading light over a small area. The glow still begins and ends sharply because the chemical reaction is controlled at the source. Larval fireflies also produce light, and their light organs have related features, though placement and function differ. This continuity shows that the basic machinery can operate in multiple life stages. In adults, the organ becomes integrated with flight and signaling, requiring faster control and higher output. A firefly flash is therefore a whole-body event expressed through a small area. Air moves through spiracles and tubes. Nerves set timing. Light-producing cells shift chemical state. Reflective layers direct photons outward. The abdomen provides a window that releases light with minimal loss. All of these structures work together so light can appear, disappear, and repeat in patterns that remain stable across a night. During flashing, the abdomen often makes small pumping movements. These movements shift internal pressure and help move air through fine tracheal branches. On still evenings, a perched adult may lift the abdomen slightly to keep the emitting surface clear of the nearby leaf. This positioning keeps the light organ unobstructed and reduces absorption by wet surfaces or dense plant tissue. Firefly reproduction begins with the placement of eggs in environments that support slow development rather than rapid change. After mating, females move close to the ground and search for areas where moisture, temperature, and shelter remain stable. Soil, leaf litter, moss, and the bases of grasses provide these conditions. Eggs are not scattered at random. They are placed where the surrounding material can buffer against drying and temperature swings. Each egg is small, soft, and enclosed in a thin shell. The shell allows gas exchange while limiting water loss. Inside, the embryo develops gradually. Cell division begins soon after the egg is laid, using energy stored in the yolk. No light is required at this stage. Development proceeds in darkness, guided by internal chemical gradients rather than external cues. Moisture plays a central role during incubation. If the surrounding environment dries too quickly, water leaves the egg faster than it can be replaced, and development slows or stops. If the environment becomes saturated, oxygen diffusion is reduced. Successful sites maintain dampness without standing water. This balance keeps gas exchange efficient while preventing dehydration. Temperature influences the rate of development, but not the direction. Warmer conditions speed cell division and tissue formation. Cooler conditions slow the process. Firefly eggs tolerate a range of temperatures, but extreme heat or cold disrupts enzyme activity. Because eggs are laid close to the ground, soil and leaf litter help moderate these changes by insulating against rapid shifts. As development continues, the embryo begins forming the basic insect body plan. Segments appear along the length of the body. Internal tissues differentiate into digestive, nervous, and muscular systems. External structures remain simple. The embryo does not yet resemble the adult form. It prepares instead for the larval stage, which will carry out feeding and growth. Some firefly eggs produce faint light. This glow is not rhythmic or patterned. It appears as a steady emission and likely results from early activation of the same chemical machinery used later in life. The light is weak and serves no known signaling function. It remains within the egg and does not attract other organisms from a distance. The presence of light at this stage does not alter development. Embryonic growth proceeds whether light is visible or not. The chemical pathway exists, but neural control is absent. Oxygen diffuses passively through the eggshell, allowing low-level reactions to occur without regulation. Incubation time varies depending on species and environmental conditions. In some regions, eggs hatch within a few weeks. In cooler climates, development may extend longer. The embryo remains dormant only if conditions become unfavorable. Once suitable moisture and temperature return, development resumes. Hatching occurs when the larva breaks through the eggshell, using small movements of the head and body. The newly emerged larva is soft and pale. Its cuticle hardens gradually after exposure to air. At this point, the larva enters a different physical relationship with the environment. It must begin feeding to support further growth. The larval body differs sharply from the adult. It is elongated and segmented, with a flattened appearance. Legs are short and adapted for crawling rather than flight. The head bears mouthparts designed for grasping and piercing prey. The nervous system is functional but simpler than in adults, supporting slow movement and basic responses. Larval fireflies are predators. Soon after hatching, they begin searching for small invertebrates, such as snails, slugs, and worms. Movement is slow and deliberate. The larva follows chemical cues and surface textures rather than visual signals. Feeding occurs close to the ground or within soil and leaf litter. Energy gained during this stage supports growth over months or even years. The larva sheds its outer cuticle multiple times as its body expands. Each molt allows internal tissues to increase in size. Between molts, the cuticle restricts growth, encouraging steady accumulation of reserves rather than rapid expansion. Larval light production may continue at low levels. The glow remains steady rather than pulsed. It may deter predators by signaling unpalatability. The light is produced by similar chemical pathways as in adults, but without the rapid control required for signaling. The abdomen contains developing light organ tissue that will later be reorganized. The early life stage is shaped by patience rather than speed. Eggs and larvae remain hidden and protected. Growth proceeds in small increments. Energy is stored rather than spent on movement or display. This strategy reduces exposure and allows fireflies to survive in environments where conditions change slowly. By the end of the larval stage, the body contains accumulated reserves that will support transformation into the adult form. The early stages therefore focus on placement, protection, and steady development. Eggs establish the foundation. Larvae expand it. The timing and conditions of these stages determine whether the firefly reaches maturity at all. As the firefly larva continues its development, daily life remains centered on the ground and the layers just beneath it. The body moves through soil, leaf litter, and decaying plant material, rarely entering open space. Movement is slow and controlled. The larva presses against surrounding surfaces, using resistance and texture to guide direction. This contact provides stability and limits exposure. The larval body is elongated and flattened, a shape that allows it to slip through narrow gaps rather than cross open ground. Short legs grip uneven surfaces and pull the body forward in small, measured steps. The head leads the movement, sweeping gently from side to side. Chemical cues on soil particles and plant debris guide this motion. Vision plays almost no role. Light near the ground is weak and inconsistent, even during daylight. Feeding dominates this stage of life. Firefly larvae are predators, targeting slow-moving invertebrates that share the same damp environments. Snails, slugs, worms, and soft-bodied insects provide reliable food sources. These prey animals leave chemical traces as they move. The larva follows these traces until contact is made. Once prey is located, the larva anchors itself using its legs and body segments. Mouthparts grasp the prey firmly. Digestive compounds are delivered that immobilize the animal and begin breaking down tissues. Digestion starts outside the body. Liquefied nutrients are then drawn in slowly. Feeding sessions can last for extended periods, during which the larva remains largely still. After feeding, the larva withdraws to a sheltered location. Movement decreases while digestion continues internally. Nutrients are distributed to growing tissues and stored as reserves. Fat bodies within the larva expand gradually, storing energy that will later support metamorphosis and adult activity. This stage prioritizes accumulation rather than expenditure. Growth is constrained by the larval cuticle. The outer covering provides protection, but does not stretch. As internal tissues increase in volume, pressure builds beneath the cuticle. When this pressure reaches a limit, the larva prepares to molt. Activity decreases. Feeding stops. A new cuticle forms beneath the old one. Molting occurs when the old cuticle splits along predetermined lines. The larva slowly pulls free, leaving the old covering behind. The newly exposed body is soft and pale. During this period, the larva remains hidden and inactive. The new cuticle gradually hardens, restoring protection. Only once this process is complete does normal movement resume. This cycle repeats multiple times over the larval lifespan. Each molt allows an increase in body size and internal capacity. With each stage, feeding becomes more efficient, and reserves accumulate more rapidly. Growth remains steady rather than fast, shaped by environmental conditions and food availability. Light production during the larval stage continues at low levels. The glow is steady and faint, without rhythmic control. It originates from developing light organ tissue that already contains the necessary chemical machinery. Oxygen reaches these tissues passively, allowing continuous low-level emission. The nervous system does not yet regulate timing. This glow may function as a warning. Many firefly larvae contain chemical compounds that make them unpalatable to predators. The faint light may signal this property at close range. The glow remains near the ground and does not travel far. It does not attract other larvae or serve a social role. The larval nervous system supports basic coordination rather than complex behavior. Responses to touch and chemical cues are reliable. Movement remains deliberate. There is no need for rapid reaction or pattern generation. Survival depends on remaining concealed, feeding efficiently, and conserving energy. Environmental conditions strongly influence larval life. Moisture is essential. Dry conditions reduce prey availability and increase water loss. In response, larvae retreat deeper into soil or beneath dense cover. Activity decreases until moisture returns. Prolonged dryness can slow growth, but rarely causes immediate death if shelter is available. Temperature also affects development. Warmer conditions increase metabolic rate and speed growth. Cooler conditions slow activity and extend development time. In many regions, larvae overwinter one or more times. During cold periods, metabolism drops and movement becomes minimal. Stored energy supports basic maintenance until conditions improve. The larval stage often lasts far longer than the adult stage. Months or even years may pass as the larva feeds, molts, and grows. Throughout this time, the body plan remains focused on crawling, feeding, and storage. Wings do not develop externally. Light organs remain simple and secondary. As the end of larval development approaches, internal changes begin. Hormonal signals shift. Feeding gradually decreases. The larva becomes less active and begins searching for a stable location. This site must remain undisturbed for an extended period. Soil chambers, rotting wood, or dense litter provide suitable conditions. Once a location is chosen, movement stops. The body prepares for reorganization rather than growth. The long period spent beneath the surface has built the reserves and structures needed for the next stage. The larva's quiet life of feeding and concealment now gives way to transformation. Energy storage during the larval stage determines whether a firefly survives long enough to transform. While feeding supplies immediate nutrients, survival depends on how those nutrients are processed, stored, and protected over extended periods underground. The larval body is organized to favor accumulation rather than speed, converting irregular meals into reserves that can sustain long intervals without feeding. After digestion, nutrients enter circulation and are distributed through the body cavity. Much of this material is directed into specialized tissues known as fat bodies. These tissues function as long-term storage rather than active organs. They expand gradually as feeding continues, filling available space between muscles and internal structures. The stored material includes lipids, proteins, and carbohydrates, each held in forms that can be accessed later. Lipids form the primary energy reserve. They pack large amounts of energy into small volumes, which is important for a body that must remain compact. Proteins are also stored, not as fuel, but as material for later reconstruction. During metamorphosis, many larval tissues will be broken down and rebuilt. Stored proteins supply the raw material for this process without requiring additional feeding. Carbohydrates are stored in smaller amounts, often converted into glycogen. These reserves provide quick access energy for basic activity and maintenance. During periods of inactivity, such as cold weather or drought, metabolism slows, and these reserves are consumed sparingly. The balance between storage and use is regulated continuously by hormonal signals. Life below ground is unpredictable. Prey availability changes with moisture and temperature. A productive feeding period may be followed by weeks of scarcity. Energy storage smooths these fluctuations. When prey becomes scarce, the larva reduces movement and feeding attempts. Stored reserves maintain essential functions, while activity remains minimal. Survival below ground also requires protection from environmental stress. Soil and leaf litter provide insulation, but conditions still vary. Heavy rain can flood spaces and reduce oxygen availability. Extended dry periods can lower humidity and increase water loss. The larval body responds by adjusting activity rather than relocating quickly. During wet conditions, larvae may remain closer to the surface where prey is abundant. During dry conditions, they retreat deeper where moisture is retained longer. Movement is slow and deliberate, conserving energy. Each relocation is weighed against the cost of movement itself. Energy storage allows larvae to wait rather than roam. Respiration continues even during inactivity. Oxygen enters through spiracles and travels through the tracheal system. When the larva is still, oxygen demand is low, metabolic rate decreases, and gas exchange slows. This reduces water loss and preserves reserves. The body enters a maintenance state rather than a growth state. Cold conditions impose further constraints. As temperature drops, enzyme activity slows. Muscle contraction weakens. Digestion becomes inefficient. During these periods, larvae often cease feeding entirely. Energy use shifts to maintaining cellular integrity rather than growth. Stored reserves supply this need without drawing attention through movement. Overwintering is a common feature of firefly development in temperate regions. Larvae remain underground through cold months, sometimes for more than one season. During this time, the body consumes reserves gradually. Fat bodies shrink slowly, but remain sufficient if accumulation during warmer months was adequate. Energy storage is not evenly distributed. Certain regions of the body accumulate more reserves than others. The abdomen, already large and segmented, expands to accommodate storage tissue. Muscles maintain minimum mass, required for posture and movement. Excess tissue is directed away from areas that must remain functional. The larval cuticle contributes indirectly to survival. By limiting water loss and providing protection, it reduces the energy cost of maintaining internal balance. A damaged cuticle increases water loss and infection risk, which raises metabolic demand. Healthy larvae therefore prioritize remaining sheltered and avoiding abrasion. Light production during the larval stage continues at a low level, but it consumes little energy. The steady glow requires oxygen and chemical readiness, but demand remains small compared to feeding and movement. Because the glow is not actively controlled, it does not fluctuate rapidly and does not impose high metabolic cost. Predators below ground rely on chemical and tactile cues rather than vision. Many potential predators learn to associate the larval glow with unpalatability. This reduces attack frequency and lowers the energy spent on escape or repair. Avoiding predation preserves both tissue and reserves. As larvae grow larger, feeding efficiency increases. Larger mouthparts handle prey more effectively. Digestive capacity expands. Each successful feeding event yields greater net gain. This acceleration allows larvae to build reserves more quickly toward the end of development, compensating for earlier, slower accumulation. Hormonal regulation coordinates storage and use. Juvenile hormones maintain the larval state while promoting feeding and growth. As reserves reach sufficient levels, hormone balance begins to shift. Signals accumulate gradually rather than abruptly. The larva does not transform until storage reaches a threshold that can support reorganization. Survival below ground also depends on timing. Entering metamorphosis too early risks running out of energy before adult structures are complete. Delaying too long increases exposure to environmental hazards and predators. The larval stage, therefore, balances patience with readiness, guided by internal signals tied to energy status. When feeding decreases near the end of larval life, energy use shifts again. Movement slows further. The larva becomes selective about location, seeking areas with stable temperature and moisture. Remaining reserves are protected rather than spent. The body prepares for a phase where feeding will stop entirely. At this point, survival depends entirely on what has been stored. The energy accumulated underground must carry the insect through transformation and into early adulthood. Every flash, flight, and reproductive effort will draw from these reserves. The long, quiet period beneath the surface determines the visible life that follows. Energy storage and survival below ground are therefore not passive processes. They involve continuous adjustment between feeding, movement, shelter, and rest. The larva does not rush. It builds slowly, responding to conditions rather than forcing change. This strategy allows fireflies to persist in environments where resources are seasonal and unpredictable. By the time the larva is ready to leave this stage, its body contains the materials required for complete reorganization. Muscles, wings, nervous systems, and light organs will all be rebuilt from what has been stored. The hidden work of energy accumulation makes the brief adult life possible. Metamorphosis in fireflies begins without a visible signal. There is no sudden change in posture or movement. After the larval stage ends, feeding stops gradually rather than abruptly. The larva becomes less active and remains within sheltered material where temperature and moisture remain stable. Soil chambers, decaying wood, and compact leaf litter provide the stillness required for the next phase. Once movement ceases, the body shifts priorities. Energy use moves away from locomotion and digestion and toward internal restructuring. This change is governed by hormones rather than external cues. Juvenile hormone levels decline slowly, allowing other chemical signals to become dominant. These signals do not initiate instant transformation. They begin a controlled sequence that unfolds over time. The larval cuticle separates from the tissues beneath it. This separation creates a narrow internal space where reorganization can proceed. Muscles that once powered crawling relax and detach from rigid control. The body becomes immobile, not because it is inactive, but because movement would interfere with reconstruction. Inside the body, many larval tissues begin to break down. This breakdown is precise rather than destructive. Cells are dismantled in a regulated process that releases proteins, lipids, and other components into circulation. These materials are not discarded. They are captured and reused. The larval body becomes a source of raw material for the adult form. At the same time, small clusters of cells known as imaginal discs begin to expand. These discs were present throughout larval life, but remained inactive. Now they receive chemical signals that trigger growth. Each disc follows a distinct path. Some will form wings, others will form legs, antennae, eyes, or reproductive organs. Growth occurs gradually, with each structure developing according to its own timing. The nervous system undergoes extensive reorganization during this period. Larval neural circuits that controlled crawling, feeding, and simple reflexes are reduced. New pathways begin forming to support flight control, light signaling, and adult sensory processing. Connections are rewired rather than added all at once. This gradual change prevents conflict between old and new control systems. Muscle tissue follows a similar pattern. Larval muscles designed for slow ground movement are broken down. New muscle fibers form in their place, aligned for wing movement and abdominal control. These fibers require precise attachment points and coordination. Building them slowly ensures correct orientation and strength. The tracheal system adapts as well. Tubes that once delivered oxygen primarily to crawling muscles and digestive tissues are reshaped. New branches form near developing wings and light organs. Oxygen delivery must remain functional throughout metamorphosis, even as internal architecture changes. This requires careful repositioning rather than replacement. Light organs continue developing during this stage. Cells that previously produced faint, steady glow reorganize into layered structures capable of rapid control. Reflective tissue forms beneath light-producing cells. Tracheal branches reposition to allow precise oxygen delivery. Neural connections begin forming that will later regulate flashing patterns. The chemical machinery remains present, but control structures mature slowly. Energy used during metamorphosis is continuous and unavoidable. Stored lipids provide most of the fuel. They are broken down gradually to support cellular activity. Proteins released from dismantled larval tissues supply material for constructing adult organs. Carbohydrate reserves support baseline metabolic needs. Because feeding has stopped, balance is essential. Energy must last until emergence. Temperature influences the pace of these changes. Warmer conditions accelerate biochemical reactions and shorten the transformation period. Cooler conditions slow cellular processes and extend development. The direction of change remains fixed, but timing adjusts to the environment. This flexibility allows fireflies to complete metamorphosis under different seasonal conditions without altering the outcome. Moisture remains important throughout this stage. Excess dryness increases water loss and raises metabolic demand. Excess moisture reduces oxygen availability. The chosen pupation site buffers against these extremes. Stable conditions allow internal chemistry to proceed without interruption or stress. There is no advantage to speed during metamorphosis. The body is concealed and immobile. Predation risk is reduced through hiding rather than escape. Errors in tissue formation cannot be corrected later. Precision matters more than pace. This is why metamorphosis unfolds gradually rather than in a rush. As development progresses, adult features become increasingly distinct. Wings fold along the body in their final configuration. Compound eyes reach full size and structure. Antennae elongate and differentiate. Reproductive organs mature internally. The abdomen reshapes, shifting space from storage tissue to signaling and reproductive structures. Near the end of metamorphosis, muscle fibers strengthen, neural circuits stabilize, oxygen delivery pathways settle into their adult arrangement. The body prepares for movement, but activity remains minimal. Systems must be synchronized before exposure to the external environment. The final stage involves hardening of the new cuticle. Structural proteins cross-link, increasing strength. Pigments develop, giving the adult body its characteristic appearance. The light organ becomes fully functional, but remains inactive. The adult form is complete, while still enclosed. When external conditions are favorable, emergence occurs. The casing splits, and the adult firefly slowly pulls free. Movement is careful and limited. Wings expand as fluids redistribute through veins. The cuticle continues to harden. The insect remains still while systems adjust to exposure. This emergence marks the end of transformation, not the beginning of full activity. Flight and signaling will occur later, once strength and coordination are sufficient. The adult body carries no margin for error. Its brief lifespan depends entirely on the success of this hidden phase. Metamorphosis in fireflies is therefore not a dramatic event. It is a prolonged process of dismantling and rebuilding, guided by stored energy and stable conditions. The absence of urgency reflects the need for accuracy. Every structure used in flight and light signaling is shaped during this time. The long, quiet transformation beneath soil and leaf litter makes the visible life of the firefly possible. Without this careful reconstruction, light would not flash, wings would not lift, and reproduction would not occur. The adult stage depends completely on the work completed here, out of sight and without haste. Emergence into the air begins cautiously. After metamorphosis is complete, the adult firefly remains still for an extended period. The body has been rebuilt, but it is not yet ready for movement. Wings are folded, muscles are newly formed, and internal fluids must redistribute before flight becomes possible. During this time, the insect stays close to the ground, often within the same sheltered location where transformation occurred. When emergence begins, the outer casing splits along predetermined lines. The adult firefly pulls free slowly, using newly formed legs to brace against surrounding surfaces. Movement is deliberate. The cuticle is still soft, and excessive force could cause damage. Once outside, the insect remains largely motionless, while exposure to air triggers further hardening of the body covering. Wings require special attention. At first, they appear crumpled and shortened. Fluids are pumped through fine veins, causing the wings to expand to full size. This expansion must occur evenly. Uneven pressure can distort wing shape and compromise flight. The insect remains still while this process completes. As the wings expand, muscles begin low-level activity. These early contractions help establish coordination rather than producing lift. The thorax warms slightly as muscle fibers engage. This warming improves flexibility and prepares the muscles for sustained movement. The process unfolds over time rather than in a single moment. The abdomen also adjusts. During metamorphosis, internal organs shifted position. Now they settle into their adult arrangement. Light organs, digestive structures, and reproductive tissues stabilize. Tracheal tubes supplying oxygen align with new muscle groups and signaling tissues. Gas exchange increases gradually as activity rises. Once the cuticle hardens sufficiently, the firefly begins testing movement. Legs flex and extend. The insect shifts position on the substrate. These small actions provide feedback to the nervous system, confirming that connections formed during metamorphosis are functioning correctly. Errors are corrected through repetition rather than rapid motion. Initial flight attempts are short and controlled. The firefly lifts briefly, then lands nearby. These movements are not displays, they are adjustments. Each short flight improves coordination between wings, muscles, and sensory input. The insect learns the limits of its newly formed body through experience. Navigation at this stage relies heavily on environmental cues. Visual information is limited near the ground, especially at dusk. The firefly uses contrast, movement of air, and surface texture to orient itself. Flight remains close to vegetation and ground features that provide reference points. Light use begins cautiously. Early flashes may be infrequent or poorly timed. Neural control of oxygen delivery to the light organs continues to refine. With each activation, feedback improves precision. Timing becomes more consistent as neural pathways stabilize. The first flashes are not yet part of full communication. They occur during short flights or while perched. The firefly tests its ability to produce light without exhausting energy reserves. Because adult feeding is limited, energy must be conserved. Each flash draws from stored reserves accumulated during larval life. Flight efficiency improves over time. Muscle fibers strengthen with use. Wing beats become smoother. The insect gains confidence in maintaining altitude and direction. Movement remains measured rather than rapid. The adult stage prioritizes endurance and precision over speed. As night deepens, the environment becomes more suitable for extended activity. Temperature stabilizes. Air movement slows. Contrast between light and darkness increases. These conditions support longer flights and clearer signaling. The firefly responds by extending activity duration. Light signaling becomes more structured. Flashes begin to follow consistent spacing and duration. This regularity allows recognition by other fireflies of the same species. The light is no longer exploratory. It becomes communicative. Positioning matters. Fireflies often fly low or perch on stems and leaves. This placement keeps signals within sight lines shaped by vegetation. Open air above allows light to travel farther horizontally. Dense foliage below absorbs excess light and reduces background interference. Energy management continues to guide behavior. Periods of flight alternate with rest, flashing pauses between sequences. These breaks allow oxygen levels to reset and chemical substrates to recover. The insect avoids continuous output that would drain reserves quickly. Predation risk shapes early adult behavior. Many predators are active at the same time fireflies emerge. Some learn to associate light with prey. Fireflies respond by limiting flash duration and spacing signals. Short, controlled flashes reduce exposure while remaining effective. The adult digestive system plays a limited role at this stage. Some species consume nectar or other small food sources, but many rely primarily on stored energy. Feeding opportunities supplement reserves, but do not replace the larval accumulation. This constraint keeps adult life relatively brief. As nights pass, coordination improves further. The nervous system refines control over wing movement and light timing. Muscles operate more efficiently. Flight paths become smoother and more predictable. The firefly's presence in the night becomes more stable. This stage marks the transition from preparation to participation. The insect is no longer rebuilding or testing. It is now part of the active night environment. Movement, signaling, and interaction occur within the limits set by energy and structure. Emergence into the air is therefore not a single event, but a gradual expansion of capability. Each step builds on the last. The body moves from stillness to motion, from silence to light, without urgency. Precision remains more important than speed. The adult firefly carries forward the work of earlier stages. Stored energy, rebuilt tissues, and refined control all converge here. The visible life of flight and light depends on this careful entry into activity. Without it, the adult stage would fail before it truly begins. Light becomes communication only when timing, position, and context align. In adult fireflies, light is not produced continuously. It appears in brief intervals that stand out against darkness. These intervals are shaped by neural control and by the physical limits of the environment. A flash carries information because it occurs at a specific moment and from a specific place. Communication begins with visibility. At night, contrast determines whether a signal can be detected. Fireflies operate when background light is low enough that a brief glow can be seen clearly. They choose heights, flight paths, and perching locations that keep their light unobstructed by dense vegetation. Open air above grasses and low shrubs allows light to travel horizontally, where other fireflies are most likely to be present. The flash itself is produced through rapid control of oxygen delivery to the light organs. When neural signals allow oxygen to reach the reaction site, light appears. When oxygen is withdrawn, light stops. This start and stop control allows precise shaping of duration. A flash may last only a fraction of a second. The brevity is not accidental. Short flashes reduce energy use and limit exposure to predators while remaining visible. Spacing between flashes matters as much as the flashes themselves. Fireflies do not flash continuously. They pause between signals, allowing each burst of light to be read independently. These pauses prevent overlap and confusion when multiple individuals are active in the same area. The absence of light becomes part of the signal by separating one flash from the next. Position in space adds further meaning. A flash produced while flying carries different information from a flash produced while perched. Movement indicates direction and distance. A stationary flash marks location. Fireflies adjust their behavior depending on whether they are searching or responding. These adjustments are guided by sensory feedback rather than conscious choice. The nervous system coordinates this behavior by integrating visual input with internal timing. When a firefly detects a flash from another individual, neural circuits respond by adjusting flight path or flash timing. This response does not require long sequences. It relies on simple timing relationships that can be repeated reliably. Communication through light depends on consistency. For a signal to be recognized, it must occur in a predictable pattern. Fireflies of the same species produce flashes with similar timing and spacing. This similarity allows individuals to distinguish relevant signals from background noise. In areas where multiple species coexist, differences in timing reduce interference. The environment shapes how these signals are perceived. Humidity, vegetation density, and air movement all influence visibility. Moist air can scatter light slightly, softening edges and reducing range. Dense foliage blocks direct lines of sight, creating pockets where signals appear and disappear as individuals move. Fireflies respond by adjusting height and movement to maintain contact. Communication is not constant across the night. Activity increases as conditions become optimal. Early evening may be too bright. Late night may be too cool. Fireflies concentrate signaling within windows where contrast, temperature, and energy availability align. This timing increases efficiency and reduces wasted effort. Light as communication also depends on the receiver's visual system. Firefly eyes are tuned to detect changes in brightness rather than detailed shapes. They respond quickly to brief flashes. The color of the light falls within the sensitivity range of these eyes, allowing detection without requiring high intensity. Matching signal output to sensory capacity reduces energy cost. Responses to received signals are not immediate reflexes. A firefly often waits for a complete flash sequence before responding. This delay prevents reacting to random light sources or partial signals. The pause allows the nervous system to confirm timing and pattern. Only then does the insect adjust its own signaling. Communication through light occurs within a broader sensory context. Chemical cues and air movement provide background information about location and activity. Light does not replace these cues. It adds a layer that works effectively in darkness. The integration of multiple senses stabilizes behavior and reduces error. Predation risk influences communication strategies. Some predators learn to associate light with prey. Fireflies respond by limiting flash duration and by pausing after signaling. The pauses reduce predictability. The brief nature of flashes lowers the chance of being targeted during movement. Energy constraints remain present throughout communication. Each flash consumes resources. Fireflies balance signaling with rest. Periods of activity alternate with stillness. During rest, oxygen demand drops and chemical substrates recover. This cycling allows sustained communication across multiple nights without rapid depletion. Communication also involves choice of distance. Fireflies rarely signal across large gaps unless conditions are favorable. Shorter distances reduce energy needed for visibility and improve accuracy. Individuals often move closer before engaging in extended exchanges. This movement is slow and deliberate, guided by repeated signals. The effectiveness of light as communication depends on repetition over time. Through repeated nights of activity, consistent patterns become reinforced. Individuals that match species-specific timing are more likely to be recognized. Those that deviate may be ignored. Over generations, this reinforcement maintains stable signaling behavior. Light communication does not carry complex messages. It conveys presence, readiness, and location. The simplicity reduces cognitive demand and error. In the dark, clarity matters more than detail. Fireflies use light to establish contact, not to transmit varied information. This form of communication is shaped by physical limits rather than choice. Darkness restricts vision. Energy limits output. Predators constrain exposure. Within these limits, light becomes an efficient tool. It appears briefly, conveys essential information, and disappears again. As the night progresses, communication patterns shift. Activity may peak and then decline as energy reserves diminish. Fireflies reduce signaling frequency and increase rest. Communication slows without stopping entirely. The night gradually returns to stillness as conditions change. Light as communication therefore operates as a controlled exchange rather than constant display. It relies on timing, spacing, and context. Each flash is shaped by chemistry, nervous control, and environment. The result is a system that functions reliably in darkness without excess. Within this system, light becomes information only because it is constrained. The limits imposed by night make brief signals meaningful. Fireflies use light not to illuminate the world, but to connect with one another across short distances under specific conditions. The communication remains simple, precise, and shaped by the physical realities of life after sunset. As communication continues through repeated flashes, distance between individuals often changes. A responding firefly may shorten the gap gradually, using light as a guide rather than moving directly toward the source. Each adjustment reduces uncertainty and improves signal clarity. The movement is slow enough to avoid sudden exposure, yet deliberate enough to maintain contact. Vegetation shapes these exchanges. Stems, grasses, and low branches interrupt straight lines of sight. Fireflies compensate by shifting position rather than increasing brightness. A small change in height or angle can restore visibility without additional energy cost. This behavior keeps signaling efficient and limits unnecessary output. Wind also affects communication. Gentle air movement can sway vegetation, briefly blocking or revealing light. Fireflies respond by extending pauses between flashes, waiting for stable moments. Timing adapts to environmental motion rather than forcing constant signaling. This restraint preserves clarity when conditions fluctuate. As multiple individuals interact, signaling becomes layered. One flash may be answered while another fades. Fireflies rely on timing rather than brightness to separate signals. Consistent spacing allows each exchange to remain distinct, even in groups. The nervous system filters incoming flashes, responding only to those that match expected intervals. Over time, communication settles into a rhythm shaped by fatigue and recovery. As energy reserves decline, flashes become less frequent. Rest periods lengthen. Activity contracts toward shorter windows when conditions are most favorable. Communication remains possible, but becomes more selective. This gradual narrowing reflects physical limits rather than decision. Muscles tire. Chemical substrates recover more slowly. The body responds by spacing effort further apart. Light remains controlled, appearing only when conditions support clear exchange. By the later hours of activity, communication slows naturally. Fewer flashes appear across the landscape. Distances increase as individuals rest. Darkness regains dominance, punctuated only occasionally by brief signals that mark continued presence rather than active exchange. Light fades again as activity pauses, leaving the night mostly still, with only occasional movement beneath grasses and low leaves. Patterns of flashing differ between firefly species in ways that remain stable across generations. These patterns are defined by timing rather than brightness. Duration, spacing, and repetition form the structure of each signal. A flash that lasts a fraction longer, or a pause that extends slightly, can mark a clear difference between one species and another. The foundation of these patterns lies in neural timing. The nervous system controls when oxygen reaches the light organs and how long it remains available. Small differences in neural firing intervals produce consistent differences in visible output. Because these intervals are controlled internally, patterns remain reliable even when environmental conditions vary. In one species, flashes may appear as single pulses separated by long pauses. In another, pairs or short sequences occur with shorter gaps between them. Some species maintain steady rhythms. Others vary timing slightly within a narrow range. These differences are subtle but repeatable. Recognition depends on this consistency rather than on complexity. Spacing between flashes often carries more information than flash length. A pause that lasts 1 second rather than 2 changes how the signal is interpreted. Fireflies attend closely to these intervals. Their nervous systems are tuned to detect expected timing and to ignore signals that fall outside that range. Color also varies between species, though within a narrower spectrum. Most fireflies produce yellow-green light, but slight shifts toward green or amber occur. These shifts result from differences in chemical environment within the light organ. Color differences alone rarely determine recognition, but they reinforce timing cues. Flash height contributes to pattern recognition as well. Some species fly higher above the ground, while others remain close to vegetation. Signals occurring at consistent heights reduce overlap between species sharing the same habitat. Height becomes part of the pattern, without requiring additional energy. Movement during flashing adds another layer. A flash delivered while moving upward differs from one delivered while hovering or perched. Direction and speed influence how the signal is perceived. Fireflies often combine movement with timing to refine recognition without increasing brightness. Environmental structure shapes how patterns evolve. In dense vegetation, shorter flashes with longer pauses reduce overlap and confusion. In open areas, longer flashes may remain visible without interference. Species adapt patterns that remain clear within their typical habitats. Recognition occurs through comparison rather than analysis. A firefly does not evaluate every possible signal. It responds when incoming flashes match expected timing closely enough. Signals that deviate are ignored. This filtering reduces cognitive demand and prevents constant response to irrelevant light. Females often remain stationary while observing patterns. Their role depends on accurate recognition rather than active display. Males adjust flight and flashing to remain visible. The interaction relies on repeated confirmation rather than single signals. Consistency over time matters more than immediate response. Errors can occur. Occasionally, a firefly responds to a signal that only partially matches its species pattern. These interactions usually end quickly when timing fails to align on subsequent exchanges. The system corrects itself through repetition rather than precision at the first contact. Patterns remain stable because deviation reduces effectiveness. Individuals that flash too quickly or too slowly fail to elicit responses. Over time, stable timing is reinforced. This reinforcement does not require learning. It emerges through successful interaction. Light pollution disrupts pattern clarity by reducing contrast. When background brightness increases, subtle timing differences become harder to detect. Fireflies respond by shortening activity windows or shifting locations. The patterns themselves remain unchanged, but opportunities to use them decrease. Temperature affects timing slightly by influencing neural speed. Warmer conditions can shorten intervals. Cooler conditions can lengthen them. Species patterns include tolerance ranges that accommodate these shifts. Recognition depends on relative timing rather than absolute values. Humidity and air movement influence visibility, but not pattern structure. Fireflies adjust position rather than timing when conditions change. This separation keeps recognition stable while allowing flexibility in behavior. Across regions, closely related species may display similar patterns with small differences. These differences reduce overlap where ranges meet. Timing shifts act as boundaries without physical separation. The night environment allows these distinctions to persist. Species-specific patterns therefore arise from interaction between neural control, body structure, and habitat. They do not require complex signaling. Simple differences repeated consistently are enough. The darkness amplifies these differences by removing competing information. Through these patterns, fireflies maintain separation while sharing space. Light becomes a reliable marker of identity because it is constrained. Timing, spacing, height, and movement work together to create signals that remain clear without excess output. Within a single species, individual fireflies keep similar timing across many nights. One insect flashes, pauses, and flashes again with nearly the same spacing each time it is active. Another nearby responds only when that spacing matches what its nervous system expects. Early adults do not begin perfectly aligned. During the first nights of flight, flashes may drift earlier or later. With repeated activity, neural control stabilizes. Oxygen delivery becomes more precise. The pause between flashes settles into a range that remains consistent across exchanges. When several species share the same space, these small adjustments matter. A firefly ignores flashes that arrive too quickly or linger too long. It remains still when timing fails to match. Only signals that repeat with the correct spacing draw a response. Over the course of an evening, this filtering separates activity into distinct rhythms, with each species maintaining its own timing window without overlap occurring. Mating in fireflies unfolds through repeated light exchange rather than immediate contact. The process begins with signaling that establishes presence and timing. A male moves through the air, producing flashes at regular intervals. Each flash is brief and separated by pauses that remain consistent across flights. The pattern does not change in response to a single signal. It repeats so it can be recognized. Females often remain stationary during this stage. They perch on stems, grasses, or low branches where light can travel outward without obstruction. From these positions, they observe incoming flashes. Recognition depends on timing rather than brightness. When the spacing matches the expected pattern, a response follows. If timing fails, no response is given. The female response is usually a single flash, delivered after a short delay. The length of this delay matters. A response that arrives too early or too late breaks the exchange. When the delay falls within a narrow range, the male detects it and adjusts behavior. This adjustment may involve changing direction or shortening flight distance. The exchange then repeats. The male flashes again. The female responds again after the same delay. Each repetition confirms alignment. The process does not accelerate. Consistency matters more than speed. Repeated confirmation reduces error and prevents wasted movement. Distance between individuals decreases gradually. The male shortens flight paths, moving closer with each exchange. Movement remains controlled. Sudden approach would increase exposure and risk. Light guides this movement step by step, allowing alignment without direct contact. Positioning plays an important role. The female often remains slightly elevated above ground. This keeps her signal clear of dense vegetation. The male adjusts flight height to keep the exchange within a stable line of sight. Small changes in height restore visibility when plants or terrain interfere. Timing remains central throughout. The male maintains the same flash pattern. The female maintains the same response delay. Deviations usually end the exchange. This strict requirement prevents cross-species mating where patterns differ slightly. Environmental conditions influence the exchange without altering its structure. Wind can move vegetation, briefly blocking signals. In response, pauses lengthen rather than flashes becoming brighter. Waiting for still moments preserves clarity without additional energy cost. Humidity and temperature affect activity length. Warmer evenings allow longer exchanges. Cooler conditions shorten them. The exchange itself remains unchanged. Only the window of opportunity shifts. Energy use is carefully managed. Each flash draws on stored resources. Repetition is efficient because flashes are brief and pauses allow recovery. Continuous output would drain reserves quickly and increase detection by predators. The measured pace keeps risk low. Predation pressure shapes behavior indirectly. Some predators learn to associate light with prey. Fireflies reduce exposure by limiting flash duration and by remaining still between signals. The exchange occurs within narrow time windows when detection risk is lowest. Once the male approaches closely, signaling often decreases. Movement replaces light as the primary guide. The final approach happens at short range, where visual contrast and physical cues are sufficient. Light has completed its role by this point. Physical contact marks the end of the signaling phase. Mating occurs without additional display. Afterward, individuals separate. There is no prolonged association. The exchange has served its purpose. Females later move to suitable sites for egg-laying. Males resume signaling or reduce activity as energy declines. The mating exchange does not alter the basic rhythm of nightly activity. It fits within it. Light exchange during mating remains simple by design. It relies on repetition, timing, and position rather than complexity. The system works because it reduces ambiguity. Each participant responds only to signals that match expected structure. Across nights and seasons, this process remains stable. Small variations occur, but the core exchange persists. Light guides approach, confirms identity, and limits error. Once contact is made, light withdraws again. Mating through light therefore reflects the same constraints seen elsewhere in firefly life. Energy is conserved, risk is managed, precision outweighs speed. The exchange unfolds slowly, shaped by darkness and by the need for reliable recognition rather than immediate action. After separation, movement slows again. The male often remains nearby, resting on vegetation while light organs stay inactive. Muscles recover from repeated flights, and oxygen demand falls. Short pauses allow chemical substrates used in flashing to return to usable levels before activity resumes. The female shifts attention to ground conditions. She tests surfaces with her legs, sensing moisture and texture. Sites that remain damp without standing water receive longer inspection. Egg laying will not occur immediately, but selection begins early. Time spent assessing reduces later movement and energy loss. If conditions are unsuitable, the female relocates gradually. Movement remains close to cover. Light is rarely used during this phase. Darkness and stillness reduce visibility to predators and conserve remaining reserves. Males resume signaling after recovery. Flash intervals lengthen as energy declines. Some individuals leave the area, while others remain within the same patch of habitat. The pattern of activity spreads out across the night rather than concentrating in a single burst. Mating exchanges do not repeat endlessly. Once energy drops below a threshold, signaling becomes infrequent. Fireflies then prioritize rest over display. This shift protects the ability to survive long enough to complete reproduction tasks that follow. Environmental conditions continue shaping behavior. Cooler air shortens activity windows. Wind disrupts visibility and extends pauses. Fireflies adjust by waiting rather than forcing interaction. The exchange remains patient. By the later hours of the night, most activity quiets. Individuals remain perched or hidden near the ground. Light appears only occasionally, marking presence rather than seeking response. The mating phase blends back into the broader rhythm of night activity, ending without emphasis or closure. With dawn approaching, remaining individuals settle deeper into cover, where rest continues until temperature and light conditions change enough to end nocturnal activity for the coming daylight hours ahead quietly. Fireflies occupy habitats shaped by moisture, temperature, and vegetation structure rather than by open space. Their presence depends on environments that remain damp through the evening and night, allowing activity without rapid water loss. Forest edges, meadows with tall grasses, wetlands, and stream margins provide these conditions. Each habitat offers a balance between cover and visibility that supports signaling and movement. Moisture is the most consistent requirement. Fireflies avoid dry ground, where dehydration risk increases and prey becomes scarce. Damp soil and leaf litter retain humidity after sunset, keeping air near the ground stable. This stability supports flight, signaling, and rest without excessive metabolic cost. Even short dry periods can reduce activity by limiting safe movement. Vegetation structure determines how light travels. Tall grasses and low shrubs create horizontal corridors where flashes remain visible without being scattered upward. In dense forest understory, trunks and branches interrupt lines of sight, shaping movement into paths and clearings. Fireflies adapt by flying along edges where contrast remains high. Temperature sets the boundaries of activity. Fireflies are active within a narrow thermal range. Warm evenings allow longer flight periods and repeated signaling. Cooler nights shorten activity windows and reduce flash frequency. Because body temperature tracks ambient conditions, muscle performance and neural timing slow as temperatures drop. Wetlands offer particularly favorable conditions. Standing water increases humidity, and surrounding plants moderate temperature changes. Fireflies often remain near the margins rather than over open water. These areas provide shelter while maintaining clear airspace for signaling. Movement stays low and controlled. Forested habitats support different behavior. Canopy cover reduces wind and light pollution, but also limits open flight space. Fireflies respond by flying lower and pausing frequently. Signals are shorter and more carefully timed to avoid interference from foliage. Human-altered landscapes can support fireflies when moisture and vegetation remain intact. Gardens with tall grasses, leaf litter, and limited artificial lighting can become active sites. Conversely, manicured lawns and paved areas reduce habitat suitability by removing cover and drying soil. Seasonal changes alter habitat quality. Spring growth increases vegetation density and moisture retention. Summer heat can reduce humidity if rainfall declines. Fireflies adjust movement and signaling accordingly, concentrating activity where conditions remain stable. Habitat choice also affects reproduction. Females select egg-laying sites based on moisture and shelter. Areas that remain damp without flooding offer the best conditions for incubation. Soil composition and organic matter influence oxygen availability and temperature buffering. Fireflies do not migrate long distances. They remain within suitable habitat patches, moving gradually rather than dispersing widely. This limited movement makes local conditions critical. Habitat loss or drying can reduce populations quickly. Environmental noise influences signaling efficiency. Wind, rain, and artificial light disrupt visibility. Fireflies respond by reducing activity rather than compensating with brighter flashes. Waiting preserves energy and reduces risk. The relationship between fireflies and habitat is therefore precise. Activity depends on small-scale conditions rather than broad landscapes. A few meters can separate suitable and unsuitable areas. Fireflies respond to these differences continuously. Habitats that support fireflies remain quiet, damp, and structured. These environments allow light to function as communication without excess output. Where these conditions persist, fireflies remain present. Where they disappear, activity fades. As night progresses within suitable habitat, fireflies adjust behavior in response to subtle changes in air and ground conditions. Humidity often rises after sunset as temperatures fall, creating a thin layer of moist air close to the surface. This layer supports sustained activity by reducing water loss during flight and rest. When humidity drops, activity contracts toward sheltered pockets where moisture lingers. Ground conditions matter as much as air. Soil that remains loose and rich in organic material retains moisture and allows gas exchange. Compacted or heavily disturbed ground dries quickly and limits oxygen movement. Fireflies respond by avoiding these areas, remaining near leaf litter, fallen wood, or dense root networks that buffer extremes. Vegetation height influences signaling range. Low grasses allow horizontal light travel, while taller plants create staggered visibility. Fireflies navigate these differences by adjusting perch height and flight path. A short rise or descent can restore a clear line of sight without increasing brightness or energy use. Seasonal rainfall reshapes habitats over time. Extended wet periods expand suitable areas, while drought contracts them. Fireflies do not relocate rapidly. Instead, populations thin where conditions worsen and persist where moisture remains stable. This slow response ties local abundance closely to long-term habitat quality. Artificial lighting alters habitat function by reducing contrast. Even low-level illumination can disrupt signaling corridors. Fireflies respond by shifting deeper into vegetation or reducing activity altogether. The habitat may remain physically suitable, but communication becomes inefficient. Habitat use therefore reflects constant adjustment rather than fixed preference. Fireflies test conditions repeatedly, responding to moisture, temperature, structure, and light. Activity concentrates where these factors align, allowing signaling, movement, and rest to occur with minimal cost over repeated nights. Microhabitat differences within the same area also shape activity. Slight depressions collect moisture longer. Shaded edges cool more slowly. These small variations create usable pockets even when surrounding ground becomes unsuitable. Fireflies move between these pockets gradually, maintaining presence without long-distance travel. This fine-scale response allows populations to persist within fragmented environments where conditions shift night by night. Such movement remains slow and deliberate, guided by contact with vegetation and ground texture rather than visual cues. preserving energy while maintaining access to suitable conditions. This gradual adjustment keeps activity stable across changing nights, without forcing rapid relocation or increased signaling effort. Local persistence depends on these small shifts rather than broad movement. Seasonal timing in fireflies follows predictable environmental changes rather than calendar dates. Activity begins when temperature, moisture, and light conditions align within narrow limits. These limits determine when adults emerge, signal, mate, and lay eggs. The timing remains consistent within regions because the same physical cues repeat each year. Temperature provides the primary boundary. Fireflies become active when evening temperatures remain warm enough to support flight and neural control. Muscles require a minimum temperature to contract efficiently. Below this range, flight becomes unreliable and signaling precision declines. As a result, activity concentrates within warm seasons and pauses when nights cool. Moisture interacts closely with temperature. Warm but dry conditions reduce activity by increasing water loss and limiting prey availability. Warm and humid conditions support extended activity. Fireflies respond by emerging earlier in the evening and remaining active longer when humidity stays high. When moisture drops, activity windows shorten. Light levels provide the final cue. Fireflies wait until daylight fades enough to create strong contrast. Early evenings with lingering brightness delay signaling. Later in the season, shorter days allow earlier activity. This shift follows changes in sunset rather than behavioral choice. These cues combine to shape seasonal cycles. In spring, increasing temperature and moisture allow emergence. Activity builds gradually as adults appear over several weeks rather than all at once. This staggered emergence reduces competition and spreads mating opportunities across time. Mid-season represents peak activity. Conditions remain stable, and most adults are present. Signaling becomes frequent but controlled. Mating exchanges occur regularly. Energy reserves accumulated during larval life support, repeated activity across many nights. As the season progresses, conditions begin to change. Nights cool, moisture patterns shift. Adult activity declines as energy reserves diminish and environmental support weakens. Signaling becomes less frequent. Rest periods lengthen. Eventually, activity stops entirely. Population cycles reflect these seasonal constraints. Fireflies do not reproduce continuously. Each generation appears within a limited window. Eggs laid during peak season develop under favorable conditions. Larvae then grow slowly across months or years, bridging the time between adult generations. Regional differences influence timing. In warmer climates, seasons extend longer, allowing prolonged activity. In cooler regions, activity windows are shorter and more concentrated. Despite these differences, the sequence of emergence, peak, and decline remains consistent. Weather events can shift timing temporarily. Heavy rain may delay emergence. Cool spells may interrupt activity. Fireflies respond by pausing rather than adapting patterns. Once conditions return, activity resumes without long-term change. Artificial light alters perceived seasonal cues by reducing contrast. Fireflies may delay activity even when temperature and moisture are suitable. This disruption shortens effective seasons without changing biological readiness. Seasonal timing therefore emerges from repeated environmental signals rather than internal clocks alone. Fireflies respond to what conditions allow rather than what time of year it is. This responsiveness keeps activity aligned with physical limits. The seasonal cycle ends quietly. Adults disappear from the night. Light no longer appears across fields and forest edges. The population persists unseen as eggs and larvae beneath the surface, waiting for conditions to return. Within a single season, nightly timing also shifts. Early in the season, adults may require warmer temperatures before becoming active, delaying emergence until later hours. As nights warm consistently, activity begins closer to sunset. This change reflects muscle readiness rather than altered behavior. Late-season nights reverse this pattern. Cooling temperatures push activity later or shorten it entirely. Fireflies respond by limiting flight and signaling to brief intervals when conditions briefly meet requirements. These intervals may last minutes rather than hours. Rainfall patterns influence these adjustments. Sustained rain increases humidity and moderates temperature, sometimes extending activity late into the season. Dry periods reduce usable nights, even when temperatures remain adequate. Fireflies adjust by compressing activity into fewer evenings. Larval development links seasonal timing across years. Eggs laid late in the season hatch under cooling conditions. Larvae enter periods of reduced growth or dormancy until moisture and temperature improve. This delay does not alter development sequence, only its pace. Overwintering integrates into the cycle. In colder regions, larvae remain inactive for extended periods. Metabolism slows, energy use drops. Development resumes when soil warms and moisture returns. This pause aligns emergence with favorable seasons rather than calendar progression. In warmer regions, overwintering may be shorter or absent. Larvae continue slow growth year-round. Adult emergence spreads over longer periods, but still follows rainfall and temperature cycles. Predation pressure also varies seasonally. Early season predators may be fewer. Later, predator populations increase. Fireflies do not change signaling patterns in response. Instead, reduced activity and shorter seasons limit exposure. Seasonal cycles remain resilient because they depend on multiple cues. If one signal shifts, others reinforce timing. Temperature alone does not trigger emergence. Moisture and light must align as well. This redundancy prevents mistimed activity. Fireflies rarely emerge when conditions would prevent successful signaling or reproduction. The cycle self-corrects through environmental feedback. As seasons change, the visible presence of fireflies fades and returns. The disappearance does not mark absence of life, only a shift below ground. Timing remains locked to physical conditions rather than behavior. These seasonal rhythms connect generations without overlap. Adults complete their role and vanish. Larvae continue unseen. The night regains darkness until conditions again allow brief points of light to appear. Seasonal timing therefore remains steady across years, shaped by repeating environmental limits that guide emergence, activity, and disappearance without variation or instruction. These limits preserve coordination between environment and life cycle across habitats and seasons within predictable annual boundaries consistently. Activity remains absent until conditions return, keeping life cycles synchronized with environmental limits rather than internal urgency, and preserving alignment between emergence, signaling, and survival across successive seasons. Bioluminescence appears across many forms of life beyond fireflies, each shaped by its own environment and constraints. In every case, light production relies on controlled chemistry, but the way light is used differs with habitat, body structure, and timing. In marine environments, bioluminescence is widespread. Many planktonic organisms produce light when disturbed. As water moves around them, mechanical contact triggers chemical reactions that release brief flashes. These flashes do not form patterns. They appear as sudden bursts that fade quickly, marking motion rather than signaling identity. The light produced by plankton spreads through water differently than air. Water scatters light, softening edges and shortening range. This limits how far signals travel, but increases local visibility. A moving predator can trigger many flashes at once, creating a cloud of light that outlines its path. This response may confuse predators or draw attention to movement rather than to the individual organism. Some marine animals use bioluminescence continuously rather than in bursts. Certain jellyfish and comb jellies emit steady light along their bodies. The light outlines form rather than timing. Movement causes the glowing outline to shift, making shape and motion visible in dark water. Control is slower than in fireflies, relying on chemical balance rather than rapid neural timing. Deep sea fish use light in more targeted ways. Some species possess light organs near the eyes or along the body. These organs may point downward, illuminating prey below, while keeping the fish hidden from above. Others use small lights as lures, moving them slowly to attract animals that respond to motion. In these environments, bioluminescence compensates for the absence of sunlight. Light becomes a tool for detection rather than communication. Signals are often steady or slowly changing because water movement and pressure limit rapid control. Neural regulation exists, but operates within slower physical constraints. On land, bioluminescence appears in fewer groups. Fungi represent a prominent example. Some species produce a continuous greenish glow along growing tissue. The light does not flicker or pulse. It remains steady across hours or days, reflecting ongoing chemical activity rather than controlled signaling. Fungal bioluminescence occurs within the cells of the mycelium and fruiting bodies. The chemistry operates continuously, supported by metabolic processes associated with growth. Oxygen availability influences brightness, but timing remains slow. The glow often appears in decaying wood or leaf litter, where fungi spread. The ecological role of fungal light is still studied. One possibility is that the glow attracts insects that assist with spore dispersal. Another is that light is a byproduct of metabolic pathways involved in detoxification. In either case, control remains limited compared to animals that signal. Bacteria also produce light under certain conditions. In these organisms, bioluminescence depends on population density. Chemical signals accumulate as bacteria multiply. When concentration reaches a threshold, light production begins. This process, known as quorum sensing, links light output to group size rather than individual action. Bacterial light is steady rather than patterned. It reflects collective metabolic state rather than communication between individuals. The glow can persist as long as conditions remain favorable. Control occurs at the level of gene expression rather than neural timing. Insects beyond fireflies also display bioluminescence. Some beetles and fly larvae produce steady light during early life stages. The glow may deter predators or serve as a warning. Like larval fireflies, these lights lack rapid on-off control and remain constant for long periods. Across these groups, the common feature is chemistry that channels energy into photons rather than heat. The differences lie in how that chemistry is regulated. In organisms without complex nervous systems, control is slow and linked to metabolism or population state. In animals with neural control, light can be shaped in time. Environmental medium strongly influences function. In water, light spreads differently, and movement triggers responses. In soil or wood, steady glow marks presence rather than timing. In air, brief flashes travel farther and allow precise exchange. Fireflies occupy a narrow position within this range. They combine efficient chemistry with rapid neural control in an environment that allows light to travel with minimal scattering. This combination supports communication through timing rather than brightness or duration. Other bioluminescent systems demonstrate alternative solutions to darkness. Each reflects what the environment allows and what the body can control. Light appears where it can provide advantage, whether by marking movement, attracting prey, deterring predators, or signaling presence. The diversity of bioluminescent life shows that light is not a single adaptation. It is a flexible outcome of chemistry, shaped by physical context. Where rapid control is possible, timing becomes meaningful. Where control is slow, steadiness becomes useful. Across land and sea, bioluminescence remains rare compared to other sensory strategies. It appears only where benefits outweigh energy cost. In each case, light integrates into existing biology rather than replacing other senses. In terrestrial environments, soil-dwelling organisms rarely use light because scattering is limited and surfaces block visibility. Where light does appear, it remains close to the emitting tissue. This restricts function to deterrence or marking rather than exchange. Across bioluminescent systems, oxygen availability limits output. Reactions slow or stop when oxygen drops. This constraint ties light production closely to respiration and environment. Because light production consumes energy, most organisms restrict its use. Continuous glow reflects unavoidable chemistry. Controlled flashes reflect selective pressure. The balance between these outcomes depends on how precisely chemistry can be regulated within tissue. Each system operates within narrow limits imposed by surroundings. In some deep sea habitats, animals carry bacteria inside light organs. The host supplies nutrients and oxygen through surrounding tissue. When oxygen flow changes, brightness changes. The light can be shaded by shutters of skin, allowing the animal to expose or conceal the glow without producing a flash fully again. Bioluminescence in the ocean operates under conditions very different from those on land. Water absorbs and scatters light, pressure increases with depth, and temperatures remain low and stable. These factors shape how light is produced and how it functions for marine organisms across the water column. Near the surface, many organisms use bioluminescence in response to disturbance. Plankton glow when water moves around them. This response does not rely on sight. Mechanical stress triggers chemical reactions that release light. The glow appears briefly and fades as movement stops. The scattering of light in water shortens its range. Instead of traveling far, light illuminates nearby space. This makes bioluminescence effective for marking motion rather than signaling identity. A swimming animal can trigger a trail of light that outlines its path. Some predators avoid this response by moving slowly. Others exploit it, using the glow to locate prey that has already revealed itself. In this environment, light becomes part of a dynamic exchange shaped by movement rather than timing. At greater depths, sunlight disappears entirely. Here, bioluminescence becomes the primary source of visible light. Many organisms carry light organs that produce steady or slowly changing glow. This glow may outline body shape or illuminate nearby space. Deep-sea fish often possess light organs arranged in specific patterns along their bodies. These patterns do not flash rapidly. They remain steady or pulse slowly. The purpose is not communication through timing, but concealment or attraction. Counter-illumination allows fish to match the faint light from above, reducing their silhouette when viewed from below. Some species use light as bait. A small glowing structure is extended away from the body and moved gently. Prey drawn toward the light come within striking range. Control over brightness matters more than timing in these cases. In the deep ocean, pressure limits rapid physiological change. Neural signals still operate, but chemical reactions proceed more slowly. Oxygen availability remains stable but low. These conditions favor steady output rather than rapid flashes. Bioluminescent bacteria play an important role in marine systems. Many animals host these bacteria within specialized light organs. The host supplies nutrients and oxygen. The bacteria produce light continuously, as long as conditions remain favorable. Brightness reflects oxygen flow rather than neural timing. Hosts can regulate light exposure mechanically. Skin folds, shutters, or reflective layers control how much light escapes. By opening or closing these structures, the animal can expose or conceal the glow without changing bacterial activity. Jellyfish and comb jellies produce light along their bodies. The glow often follows muscle contractions, spreading across tissue as waves. Timing reflects movement rather than signaling. The effect outlines shape and motion, making the animal visible in otherwise complete darkness. Some squid eject clouds of bioluminescent fluid. This fluid glows independently, creating distraction while the animal escapes. The light persists briefly, marking a false target rather than the body itself. Throughout the ocean, bioluminescence integrates into feeding, defense, and concealment. It rarely serves long-distance communication. Water limits range and distorts timing. Steady or slow light works better than rapid pulses. Energy cost remains important. Producing light consumes metabolic resources. Marine organisms restrict output to situations where benefit outweighs cost. Continuous glow reflects unavoidable chemistry or constant bacterial activity. Controlled exposure reflects behavioral choice. The depth of the organism shapes light color. Blue light travels farthest in water, so most marine bioluminescence appears blue or blue-green. This color choice reflects physics rather than preference. Bioluminescence in the ocean demonstrates how environment shapes function. Where water absorbs light, brightness and steadiness matter more than timing. Where pressure limits rapid control, chemistry dominates regulation. These systems operate without urgency. Light appears when conditions demand it, and disappears when they do not. The ocean remains mostly dark, punctuated by brief or steady glows that serve immediate physical roles. In shallower coastal regions, bioluminescence often appears near the seafloor. Sediment disturbance triggers light from organisms living just beneath the surface. Footsteps, currents, or feeding activity produce brief glows that mark contact rather than intention. Coral reef environments show limited bioluminescence because complex structures block light and scattering reduces effectiveness. Where light does appear, it remains local and steady. Timing plays little role. Across depths, the chemistry remains similar. Light production depends on luciferin, luciferase, oxygen, and energy supply. Differences arise from regulation rather than from basic mechanism. Marine organisms rarely use light for species recognition. Distance and scattering reduce precision. Instead, light supports immediate physical interaction. Seasonal changes in ocean conditions influence visibility. Phytoplankton density alters light absorption. Turbulence changes scattering. Bioluminescent responses adjust automatically as physical conditions shift. At depth, temperature remains stable, but nutrient availability varies. Light production reflects metabolic state rather than behavioral timing. Unlike terrestrial fireflies, marine organisms do not rely on repeated exchanges. One event often completes the function. A flash marks motion. A glow attracts or conceals. Once the action occurs, light stops. This simplicity reduces error in environments where signals degrade quickly. Light remains useful only when it aligns with immediate physical needs. Across the ocean, bioluminescence persists because it fits these constraints rather than overcoming them. The chemistry endures, while the expression remains limited by environment and structure. Light therefore appears where conditions allow and disappears when those conditions no longer support it reliably. At mid-depths, slow currents transport bioluminescent organisms across large areas. Light appears sporadically as bodies contact particles or each other. These brief glows reveal density rather than identity. Movement patterns become visible in fragments, allowing predators and prey to sense activity levels without sustained illumination or repeated signaling. This partial visibility shapes behavior while keeping energy use low in deep, dim environments where conditions change slowly over long distances and time frames. Human observation of bioluminescence unfolds slowly, shaped by patience rather than intervention. For most people, encounters with living light occur by chance, often remembered more for the conditions surrounding them than for the organisms themselves. Darkness, still air, and quiet attention create the circumstances where bioluminescence becomes visible. On land, fireflies are often noticed only after the eyes adjust. At first, the night appears empty. Gradually, small flashes emerge near the ground or along vegetation edges. These flashes are brief and isolated. They do not demand attention. They appear, disappear, and reappear elsewhere, encouraging stillness rather than pursuit. Observation changes behavior. People who remain still see more. Movement disrupts sight lines and disturbs insects. By standing quietly, the observer allows flashes to occur without interruption. Over time, patterns become apparent, not because they are analyzed, but because they repeat. The surrounding environment shapes perception. Tall grasses block distant signals. Open fields allow wider view. Trees frame darkness into smaller windows where light stands out sharply. Moist air carries sound and scent, adding to the sensory field without competing with sight. Human perception is limited compared to that of fireflies. Eyes respond slowly to changes in brightness. Peripheral vision detects flashes more easily than direct gaze. Observers often notice light at the edge of vision before recognizing its source. This limitation encourages scanning rather than fixation. The pace of observation aligns with the pace of the organisms. Fireflies do not flash continuously. They pause. The observer waits through these pauses, learning that absence does not mean disappearance. Light returns when conditions align again. In marine environments, observation differs, but still depends on restraint. Bioluminescence appears when water is disturbed. Divers learn to move slowly. Sudden motion creates clouds of light that overwhelm detail. Gentle movement reveals individual responses. Dark adaptation remains essential. Artificial light destroys sensitivity. Observers shield lights or avoid them entirely. As vision adapts, faint glows become visible. The water remains dark, but movement leaves traces that fade gradually. Scientific observation follows similar principles. Researchers record timing, location, and conditions rather than forcing responses. Instruments measure light output, but fieldwork still depends on waiting. The organisms control when light appears. Observation respects that control. Long-term observation reveals cycles. Nights differ. Some evenings remain quiet. Others show widespread activity. Seasonal changes shift timing and density. Without rushing, these variations become familiar. Cultural responses to bioluminescence often emphasize wonder, but direct experience remains subdued. Light appears briefly and leaves no residue. The memory holds the context rather than the image itself. Human presence can disrupt observation. Noise, vibration, and light interfere with signaling. Effective observation minimizes these disturbances. Silence becomes part of the method. The act of watching slows perception. Attention spreads across space rather than narrowing. This broad focus matches the way bioluminescence appears, scattered and intermittent. Over time, observers learn to anticipate without expectation. Light may appear, or it may not. The night remains the same regardless. This acceptance supports continued observation without frustration. Observation does not change the organisms. Fireflies flash according to their own constraints. Marine organisms glow in response to movement. Human watching does not alter these processes unless interference occurs. By remaining passive, observation becomes participation in the environment rather than disruption of it. The observer shares the night without imposing structure. The quiet nature of bioluminescence makes it unsuitable for spectacle. It rewards those who wait and notice, rather than those who search aggressively. This form of observation reflects the broader relationship between humans and natural systems. Understanding comes through repeated exposure, not extraction. Living light does not announce itself. It requires darkness and patience. Observation aligns human behavior with environmental rhythms rather than overriding them. In this way, bioluminescence becomes a reminder of scale. The light is small, the night is large. Observation occupies the space between them, shaped by stillness and time. Historical accounts of bioluminescence often describe confusion at first encounter. Early observers mistook glowing insects or waves for distant fires or reflections. Only with repeated exposure did the biological source become clear. These accounts emphasize uncertainty rather than explanation. Modern observation benefits from accumulated knowledge, but experience remains similar. Knowing that fireflies or plankton produce light does not make the appearance predictable. Each encounter still depends on conditions and timing beyond control. In both land and sea, observation remains temporary. Light fades, organisms move on. The environment returns to darkness. Records capture fragments, but the full experience remains bound to presence. This limitation shapes scientific and personal understanding alike. Observation accepts incompleteness. Patterns emerge over time, but individual moments remain isolated. The night therefore remains primary. Bioluminescence does not transform it. It punctuates it briefly. Observation respects that balance, allowing light to appear without expectation or demand. Repeated observation also reveals absence. Many nights pass without visible light. These gaps are informative. They reflect changes in temperature, moisture, and activity. Absence becomes part of the pattern rather than a failure to observe. Observers who return over long periods develop familiarity with place rather than event. Fields, shorelines, and forests become known through accumulated quiet experiences. Light appears within this familiarity rather than standing apart from it. Such familiarity reduces urgency. Observation continues whether light appears or not. The environment remains the subject, not the spectacle. Over time, this approach fosters attention that remains open rather than fixed, allowing brief signals to register without dominating the experience of the surrounding night environment. Observation becomes an ongoing relationship with place, shaped by repetition, quiet conditions, and respect for processes that unfold without regard for human schedules or intent. In this way, watching remains patient, indirect, and grounded in physical presence. The night accommodates observation without change. It remains unchanged throughout time. As the night progresses towards stillness, activity across the landscape slows. Flashes appear less frequently and then stop altogether. Fireflies settle into vegetation or drop closer to the ground, where movement becomes minimal. The air cools and moisture gathers, softening sound and motion. Darkness regains continuity as brief points of light withdraw. This quiet phase is shaped by the same constraints that governed earlier activity. Energy reserves have limits. Muscles tire. Chemical substrates recover more slowly. The body responds by reducing output rather than forcing continuation. Light ceases not because of decision, but because conditions no longer support efficient signaling. Movement during this period becomes careful and infrequent. Fireflies remain sheltered within grasses, leaf litter, or low branches. Legs maintain contact with surfaces. Wings rest folded along the body. Oxygen demand falls as metabolism shifts toward maintenance rather than action. The environment continues to change gradually. Temperature stabilizes near ground level. Air movement declines. Insects that were active earlier retreat into cover. Predators adjust their own behavior as visibility decreases further. The night becomes less dynamic and more uniform. Light pollution, when present, remains constant rather than episodic. In its absence, natural darkness deepens evenly. Stars and moonlight provide faint background illumination, but contrast remains low near the ground. Under these conditions, firefly light would no longer stand out clearly, even if produced. The reduction of activity allows recovery. Internal chemistry resets. Oxygen levels equalize. Neural firing slows. These processes prepare the body for rest rather than immediate future signaling. The adult stage does not reverse course once activity declines. It follows a single arc shaped by limited reserves. Over successive nights, this pattern repeats with decreasing intensity. Early nights of activity give way to shorter periods of signaling. Eventually, flashes disappear entirely from the area. The population persists only in earlier life stages hidden below ground. Eggs rest within soil and plant material. Larvae continue slow growth beneath the surface, insulated from rapid change. Their world remains damp and dark, shaped by contact rather than light. The cycle continues without visible presence. The night landscape returns to a state that appears unchanged. Fields, forests, and wetlands remain in place. The absence of light does not indicate absence of life. It reflects a shift in scale and timing rather than loss. This return to stillness completes the cycle of visibility. Light appeared briefly, served its function, and withdrew. Darkness resumes its role as the dominant condition. The environment holds the memory of activity without retaining its marks. Seasonal change will eventually alter conditions again. Temperature and moisture will rise. Larvae will complete development. Adults will emerge when constraints align. The pattern will repeat, not because it is remembered, but because physical conditions allow it. For now, the night remains quiet. Motion is limited. The ground holds what cannot be seen. The cycle continues beneath the surface, waiting for conditions to shift once more. Time continues to pass without visible markers. Hours blend together as environmental cues replace activity as the primary signals of change. Cooling air thickens near the surface. Dew forms on leaves and stems. These changes occur regardless of the presence or absence of light-producing organisms. Within resting fireflies, internal processes continue quietly. Cellular repair proceeds. Waste products are cleared gradually. Stored energy is conserved rather than spent. The body remains alive and responsive without expressing outward signals. Other organisms share this stillness. Frogs reduce calling. Birds remain silent. Small mammals limit movement. The night shifts from interaction to maintenance across many forms of life. The absence of sound and light reflects synchronized restraint rather than emptiness. This phase carries no emphasis or conclusion. It is simply a return to baseline. Fireflies do not mark the end of activity. They cease when conditions no longer support it. The environment accepts this change without response. By morning, light will return from the sun rather than from living bodies. Daytime processes will resume. Fireflies will remain hidden, conserving what remains of their brief adult stage. The cycle of visibility will remain paused until the next suitable night. The story of fireflies therefore ends not with an event, but with withdrawal. Light fades, movement slows, and the night continues unchanged. This withdrawal does not erase what occurred. It simply places it back into the broader rhythm of time. Fireflies appeared when physical conditions allowed signaling to be effective. They retreated when those same conditions no longer supported clarity or efficiency. Nothing in the environment records the exchange. There are no lasting traces on leaves or soil. The only continuation lies in development occurring out of sight. Larvae remain active in their own slow way, shaped by contact and chemistry rather than light. The night holds this transition without emphasis. Darkness persists as a stable background. It supports cycles of appearance and absence without interruption. In this way, fireflies belong to the night without defining it. The landscape does not wait. It continues to respond to weather, season, and time. Fireflies move through the system briefly, contributing light for a short interval before returning to stillness. The cycle closes quietly, leaving no marker beyond memory and expectation. Night settles fully, carrying forward processes that remain unseen but continuous, ensuring that life persists through balance rather than display. and through timing rather than permanence. In this stillness, the system resets without urgency, maintaining continuity across hours, days, and seasons as conditions slowly shift again. The night remains present, quiet, and unchanged. Activity will return only when physical limits allow it. Until then, darkness remains complete. The cycle waits, quietly and without signal, still present. Thank you for listening to the Sleepy Mind channel. I hope you have enjoyed spending time learning about fireflies.